So today we're going to switch to prokaryotic gene expression. Okay, and although that is quite wide and you've done a lot of it in your first year, so what we're going to only focus on is the concept of operons. Okay, so we'll introduce what are operons, and we're going to look specifically at 3 different operons. Okay, and how they are regulated. Each one of them is regulated in a different way. So we'll look at how that is done. Okay, all right. Okay then, so operons were first identified by these 3 people, Jacob, Monod, and Lwoff. Okay, those of you who are doing biochemistry, you probably heard of at least Jacob and Monod. But essentially, an operon is a group of genes that are transcribed at the same time. Okay, and the proteins that are coded by these group of genes control one important biochemical process. All right. So basically, what this means is that you have a group of genes, which may be 3 genes or 4 genes or 5 genes, which are next to each other. Each one of them is regulated by a single promoter. So one promoter will regulate the expression of 3 genes or 4 genes or 5 genes. Okay, and so for example, if you have an operon with 3 genes, remember we're talking now about bacteria. Okay, so each gene is going to code for 1 protein, and the 3 proteins have to work together to produce one product or one biochemical process. Okay, so if only 2 are present, then you don't have the eventual product, or you don't have the biochemical reaction taking place, right? So you need to ensure that all 3 proteins are produced together, and that's why they are controlled by a single switch or a single promoter. Okay, and operons are only found in prokaryotes. You will never find operons in eukaryotic cells. Right? So this is just a simple cartoon telling you what I've just said. So if you can see here, you have 1 gene, 2 genes, and gene number 3. Okay? So gene number 1 will produce polypeptide number 1, which is folded into protein number 1. And then gene number 2 will produce the second polypeptide, which becomes protein number 2, and so on. Okay, but to switch on the expression of gene number 1, gene number 2, and gene number 3, there is only 1 promoter. Okay, remember, if it is a eukaryotic cell, then you have 1 promoter for gene 1, 1 promoter for gene 2, 1 promoter for gene 3, right? Okay, but this is a bacterial cell. This is the operon system. So 1 promoter controls the expression of many genes. Okay, so it was first found by, or first described by Jacob, Monod, and Lwoff. Okay, and the operon types, you have 2 types of operons. One is an inducible operon, one is a repressible operon. So again, what does this mean? Okay, what did I say just now? The end product of the operon gene expression, okay, is to enable a biochemical process, right? So you should get some sort of reaction taking place. Okay, so the product of that reaction can either be the inducer of the operon, or it can be the repressor of the operon. Okay, and all operons can be negatively regulated, or they can be positively regulated. Okay, all right. So I mentioned right at the beginning that you're going to be looking at 3 different operons. So we're going to start off with the first one, which is the lactose operon. Okay, and then next Monday, we will be looking at the arabinose operon. and the following lecture will be on the tryptophan operon. Okay, so for today, let's just focus on the lactose operon, or in short, it's called the Lac Operon. Right? So first of all, in terms of the structure, we'll look at the picture of this. Okay, in terms of the structure, there are 3 genes. These genes are called Z, Y, and A. Okay, so if you're asked to list down the genes that are found in a Lac Operon, you would list them as Lac Z, Lac Y, and Lac A. Okay, now in addition to the 3 genes, you have 1 promoter and 1 operator. All right, so again, recall that the operator is where the repressor is going to bind. Right? So in a eukaryotic cell, what we have seen already, the operator is the equivalent of the silencer domain. Okay? So repressors in bacterial cells will bind to operators. Repressors in eukaryotic cells will bind to the silencers. Okay? All right. So the Lac, in addition to these 3 genes, Z, Y, and A, there is another gene which is called Lac-I. Okay? Now the Lac-I gene produces the repressor molecule. So in other words, okay, this operon produces a repressor molecule, okay, to switch off the operon. Right? When the operon is not needed, then you need to be able to switch it off. And to switch it off, you need a repressor molecule. The repressor molecule is produced by the operon itself. Okay, but there is a separate promoter that controls the expression of the Lac-I gene or the repressor gene. Okay, all right. Okay, all this you can read later, but it'll become clearer once we look at the mechanism. Okay, now the Lac Operon, the biochemical reaction that takes place by the enzymes that are produced by Lac Z, Lac Y, and Lac A, okay, the overall process is to hydrolyze lactose into galactose and glucose. Okay, so let's take one step back. All right, now you've already learned about metabolism in eukaryotic cells and in prokaryotic cells. All right, the purpose of doing metabolism is to produce energy in the form of ATP. Okay, now in bacterial cells, the substrate, the carbon source, okay, to be metabolized and produced ATP, the first choice is glucose. Okay, if there is enough glucose in a bacterial cell, if the bacterial cell is growing in an environment where there is enough glucose, the bacterial cell will always use glucose first. Okay, break down the glucose to produce ATP. Okay, however, if glucose is not enough or is missing from the environment in which the bacterial cell is found, then it has to use some other sugar, which is normally a more complex sugar. Okay, and when it uses a more complex sugar, the reaction that takes place is that it must break down the complex sugar to produce glucose. Okay, or produce the ATP directly. So with the Lac Operon, what it does is the 3 enzymes are involved in breaking down lactose, which is a more complex sugar, okay, to produce galactose and glucose. Okay, so this glucose will then be used up by the bacterial cell to produce energy. Okay, and the enzyme that hydrolyzes lactose to galactose and glucose, basically hydrolysis is you're breaking this bond here. Okay, so to produce 2 separate molecules, and this reaction is catalyzed by beta-galactosidase. Okay, beta-galactosidase is the product of the Lac Z gene. Okay, remember lactate is one of the 3 genes. Okay, so to recap what I've just said, E. coli or any bacteria can use either glucose, which is a monosaccharide, or lactose or any other complex sugar or disaccharides. Okay. However, if it's going to use the disaccharide, it needs to hydrolyze it first. So hydrolyze means to break it down. Okay. So whenever glucose is present, the bacteria will prefer to use glucose. Why? Because there's no need to hydrolyze it. Doing any process requires energy, requires time. Okay, so given a choice, the bacteria will always go for no need to use energy, no need to waste time. And what is that? That is use glucose first. Okay, right. So if a bacteria cell is present in an environment where glucose is also present, okay, the E. coli will not switch on the Lac Operon. Why? Because remember, what is the purpose of switching on the Lac Operon? Is to produce the beta-galactosidase enzyme, which is needed to hydrolyze the disaccharide to glucose. Okay, but now that glucose is there, is available, there's no need to switch on the Lac Operon, right? Okay. When glucose is present and lactose is also present, so now the bacteria is in a rich environment, it's got 2 choices. It's got glucose, it's got lactose. Okay, so what does it do? It will always go for the glucose first, right? So there is no need to switch on the Lac Operon. So the Lac Operon is switched off, right? Then we have another situation where glucose is absent, lactose is also absent, okay? And again, the Lac Operon is switched off, right? Okay, you would think, now there's no glucose, okay, so we must try and switch on the Lac Operon so that we can hydrolyze lactose and produce the glucose, which the bacteria cell needs, right? But unfortunately, there is no lactose. So the lactose now is actually the inducer molecule. Okay, it is what switches on, the presence of the lactose will switch on the Lac Operon when glucose is absent. But now when lactose is also absent, so there's not going to be any transcription of the Lac Operon. Okay, the Lac Operon is only switched on when glucose is absent and lactose is present. Okay, right? Okay. So how does this happen is what we're going to be looking at next. Okay, so the structural enzymes I told you just now, okay, are encoded by the genes LAC-Z, LAC-Y, and LAC-A. Okay, you've already been introduced to LAC-Z. LAC-Z encodes beta-galactosidase. Okay, so what about LAC-Y and LAC-A? So LAC-Y encodes for this enzyme called lactose permease, right? And LAC-A encodes for an enzyme called trans-acetylase. Okay, what is the function of lactose permease? Lactose permease, its role is to bring the lactose, which is in the environment, into the bacteria cell. Okay, because the hydrolysis of lactose into galactose and glucose takes place inside the bacteria's cytoplasm. All right, but the lactose is found outside the cell in the environment. So you need something to transport the lactose from the environment into the bacterial cytoplasm. Okay, and the agent to transport the lactose is lactose permease, right? Okay. All right, so this is what the structure looks like. Okay, so you have seen all of this that we are going to be pointing at now. All right, so promoter. The promoter is always next to the operator. in a prokaryotic gene structure. Okay, all right. Downstream of the operator, you will find your Lac Z, Lac Y, and Lac A. Okay, and it has to be in this order: Z first, Y second, and A last. Okay, so Lac Z encodes for beta galactosidase, Lac Y encodes for permease, and Lac A encodes for trans-acetylase. Okay, now upstream of the promoter is the Lac I. And remember, Lac I encodes for your repressor molecule. Okay, and Lac I has a constitutive promoter. I think you all know what constitutive means, right? That means it's switched on all the time. So Lac I encodes for the repressor. The repressor is produced at low levels of expression all the time in the bacteria cell. Okay, all right? Because in a bacteria's lifetime, 90% of the time, probably about 90% of the time, no need to switch this on. You want to switch it off. Okay? And that's why you always need the Lac I repressor protein present. The only 10% of the time, 10 to 15% of the time when you need to switch this on is when suddenly the bacterial cell has no more glucose. Okay? All right. So the mechanism of how this operon, Lac Operon works, right, is, as I said just now, the repressor molecule, which is this purple molecule now, okay, is constantly being produced by the Lac I gene. Okay, so what happens is that the repressor molecule is going to go and bind to the operator. Okay, right? So you can see this, the repressor molecule in this drawing is drawn in such a way that there is a rectangle right at the top here, okay, to fit nicely into this rectangle space in the operator. So it's just like enzyme and substrate lock and key mechanism, okay, it will fit nicely. All right, so what happens is when this repressor molecule goes and binds to the operator here, okay, it is going to be blocking the promoter. Okay, so by blocking the promoter, it's going to block RNA polymerase from binding to the promoter. So if RNA polymerase cannot bind to the promoter, it cannot switch on transcription of Z, Y, and A. Okay, does everyone understand that? Understand or not?
Yes, Prof.
Okay, now let me make it clear. All right, we're going to be looking at 3 operons. As I said earlier, the Lac operon is the easiest operon for you to understand. Okay, as we move on to the ARA operon and the tryptophan operon, the regulation becomes more and more complicated. So please pay attention. When I ask a question, I expect to hear answers. Okay? All right. So now in the case where, let me move on here. In the case where the situation now is the bacterial cell, okay, has got no glucose. However, in the environment, lactose is present. Okay, so as I said earlier, the lactose permease enzyme will go and bring the lactose into the bacterial cell. Okay, now the lactose that is transported into the bacterial cell is known as allolactose. Right? So what does the allolactose do? The allolactose is now going to bind to the repressor molecule. Okay, so this is the inducer. The inducer is your allolactose. Allolactose is going to go and bind to the repressor molecule, which is Lac I. Okay, now when the repressor binds to the allolactose, what happens to the repressor molecule is what we called conformational change. Okay, conformation refers to structure. So if you can see from this diagram now, what I described to you earlier, right? I said that the repressor has got like a rectangular shape up here so that it can fit nicely into the rectangular space in the operator. Okay, now when the allolactose is present and binding to the repressor molecule, the repressor changes shape. So you can see it is up here, it is no longer rectangular. It looks more oval shape now. Okay, so now because the allolactose is binding to the repressor molecule, there is a conformational change to the structure of the repressor molecule, and the repressor can no longer bind to the operator because it doesn't fit nicely into the space in the operator. Okay, so when there is no repressor molecule sitting at the operator, the promoter is now free and open for RNA polymerase to come and bind. Okay, so when RNA polymerase comes and binds to the promoter, then it will switch on transcription of Lac Z, Lac Y, and Lac A. Okay, Lac Z, when it's transcribed and translated, will produce beta-galactosidase. Lac Y, when it's transcribed and translated, will produce permease. And Lac A will produce trans-acetylase. Okay, beta-galactosidase is to hydrolyze lactose. Permease is to transport lactose from outside the cell into the cell. Okay, and trans-acetylase, what does it do? It adds acetyl groups. Okay, why the need for these added acetyl groups? At the moment, nobody knows. All right, okay? Okay, so now I have a question for you. Okay, the question is, sorry, if the Lac Operon is repressed, okay, and no permease, which is the enzyme that is that needs to be transported, that needs to transport lactose into the cell, is being produced, how does lactose get into the cell to inactivate the repressor and turn on transcription? Okay, does everyone understand that question?
No.
No?
No, I don't understand the question.
Okay, so coming back, let me use this again. Let me see if I can find, okay. So let's wait on. Okay, let's use this. All right, let's use this picture. So remember now, the situation is, okay, the situation is, let me give you an example. I want you to imagine an E. coli cell, okay, and inside the E. coli cell, there is high concentration of glucose. Okay, so when there's high concentration of glucose but no lactose present, all right, this is just to recap, okay, so your repressor molecule is going to go and bind to the operator here. Okay, RNA polymerase cannot bind to the promoter. This is going to switch off the Lac Operon. All right, okay, now you imagine the same bacteria cell, okay, where inside the cytoplasm and in the environment, there is no glucose. There's no more glucose. Okay, so if there is no glucose, the bacterial cell cannot generate energy. Sooner or later, it's going to die. It's going to starve and then it's going to die, right? So how can it survive? The way to survive is to switch on the Lac Operon, right? And then produce the 3 enzymes to go and hydrolyze lactose into galactose and glucose. Okay, all right. But remember this, okay? At the point where there is no more glucose inside the bacterial cell, the Lac Operon is still switched off because there is no lactose inside the cell. Okay, the lactose has to be brought from outside the cell into the cell. Understand so far? Okay, But you know, to bring the lactose from outside the cell into the cell, what do you need? You need permease. Permease is coded by Lac1. Okay, that means you need to switch on the operon in order to produce permease. Permease can then go out, look for lactose, and bring it in. Right? Okay, and then beta galactosidase will hydrolyze it. Right now. But for it to be switched on, you need the lactose to already be in the cell in the form of allolactose so that it binds to the repressor, and the repressor cannot bind to the operator. RNA polymerase can bind and switch on, right? Isn't it? So it's just like the chicken and egg story, right? Okay. Did the egg crack to produce the chicken, or did the chicken produce the egg first? Which one is it? Okay. So the question is, all right, to switch on, you need lactose. To bring in lactose, you need permease. Okay, so if you haven't switched it on yet, how was there permease that brought in the lactose into the cell to bind to the repressor and stop it from binding to the operator? Okay, so to answer the question, you need to know about this concept called leaky promoter, which I'm not sure whether you've heard of it before. Okay, leaky promoter means that the promoter here, this promoter, is never completely shut off. Okay, there is still a little bit of transcription of the Lac Operon taking place all the time. It is not constitutive like the promoter for the repressor gene. Okay, it is just leaky. Leaky means it is not 100% shut off, even though the repressor molecule is binding to the operator. Okay, maybe about 2% is still switched on. And that 2% is enough to produce a small, tiny amount of permease enzyme. Okay, so when there is no glucose inside the cell, then this permease, this small amount of permease will go out into the environment, okay, will be secreted by the E. coli cell, go and search for the lactose, and then carry the lactose back into the cell. All right, so when the lactose has come in, the lactose will go and bind to the repressor. The repressor now cannot bind to the operator, all right? So RNA polymerase can keep binding to the promoter and keep switching on Z, Y, and A at very high expression levels to produce quickly beta-galactosidase, okay, which will hydrolyze the lactose to produce galactose and glucose, okay? It will produce a lot of permease to keep going out, bring some more lactose in, keep going out, bring some more lactose in, and so on, all right, to increase the concentration of the lactose, which will be kept being hydrolyzed by the beta-galactosidase, okay, until the concentration or the amount of glucose starts to increase and get higher and higher and higher, okay? As the bacterial cell can now detect high amounts of glucose, then it will start to switch off the Lac Operon. Okay, because there's no need to bring in any more lactose. There's no need to break down any more lactose because now there's enough of glucose. Okay, so if you look at the text, right, repression never completely shuts down transcription, even when repressor is bound to the operator. There's always low level of transcription, okay, and a few molecules of beta-galactosidase, permease, and transacetylase are synthesized. When lactose appears in the environment, the permease, okay, which is present in that small amount, okay, will go and pick up the lactose and bring it into the cell, right? So some of the beta-galactosidase which was present from the leaky promoter transcription, right, will convert the lactose into allolactose. Allolactose will bind to the repressor molecule, and this will start the transcription of the lactose operon, full-time, 100% transcription, until all the lactose can be broken down to produce glucose. And then the cell becomes happy because now it has a lot of glucose. There's enough of glucose, right? There's no need to switch on the Lac Operon anymore. Now, no lactose will be brought into the cell. So the concentration of lactose gets less and less and less and less. The repressor becomes free. There's no more allolactose binding to the repressor. So it's back to the original shape. Now it can bind to the operator, and that will switch off the lactose operon. Okay, right? Everyone understand or not?
Yes,
Is it clear now? Okay, so let's just go through very quickly again. Okay, so your repressor produced by the LAC-I gene. Okay, repressor goes and binds to the operator. Okay, so repressor binding to the operator is going to block transcription because RNA polymerase cannot bind here. So there's no transcription of LAC-Z, LAC-Y, LAC-A. No enzymes are produced. Okay. All right. However, when there is no glucose present, all right, then the low levels of permease will go and bring in your lactose. The lactose will, or the allolactose will bind to the repressor molecule. Now the repressor, you can see that the shape has changed from this shape, now became this shape, okay? Because it has undergone this conformational change, this one, all right, in the protein structure. It can no longer bind to the operator. When there's no binding here, now RNA polymerase is free, okay, to come and bind here. Switch on, you get transcription of 1, 2, 3 genes, produce 1, 2, 3 proteins, which will do their function, and the end product is increasing amounts of glucose. Okay? All right. Okay. So these slides I'm going to skip. All right, they're just experimental information on how the LAC-I gene was first identified and the role of the LAC-I protein as a repressor. Okay, so that's left to you to go and read it yourself. Okay, all right. So what we're going to be looking at next, okay, is you have also bacterial cells that may have undergone mutations in the Lac Operon structure, the gene structure. Okay, so what we want to see now is what happens to the Lac Operon if it has one or more mutations. So the first example is if you have a LAC-I mutant. Okay, so the genotype is written as LAC-I minus or LAC-I negative. Okay, so the negative sign is indicating that it's a mutant. So now the LAC-I gene is mutated. So what happens? Okay, so when the LAC-I gene is mutated, it produces a mutant repressor. Okay, so again, you can see that the shape of the mutant repressor is different from the normal or wild type repressor molecule. Okay, so now the mutant repressor, because of the different shape, it doesn't fit into the operator site, so it cannot bind to the operator. Okay, so when it cannot bind to the operator, this means that the promoter is free. RNA polymerase will come and bind to the promoter and switch on transcription of Z, Y, and A. Okay, so the enzymes, the three enzymes will be produced constitutively. They will be produced even though there is glucose in the cell. Do you understand? Okay. Even if glucose is present in this bacterial cell that has a Lac I mutant, okay, the repressor molecule or the Lac I molecule now cannot turn off transcription of the Lac Operon. Therefore, the Lac Operon will be always switched on, okay, right? Whether it needs glucose or doesn't need to hydrolyze lactose, they will be producing all the three enzymes. So basically, it's a waste of time, waste of energy. Okay, right. The next mutation is known as LAC-IS. Now, LAC-IS stands for super repressor mutant, right? Okay, so you can already guess super repressor is probably going to repress all the time, even in the absence of glucose. Okay. So what happens with the LAC-IS mutant is now the mutation is on the opposite side. It's on the side where the inducer or the allolactose is supposed to bind. So in fact, what happens now is in the presence of lactose or allolactose, okay, there's not enough glucose. The lactose now cannot bind to the super repressor because the shape has changed. Okay, so the inducer cannot bind to the repressor. The repressor will continue to bind to the operator, and you will switch off transcription completely. Okay, even though there is no glucose, but lactose is present, right? Unfortunately, the lactose now cannot bind to the repressor to stop it from switching off the operon. Okay, so the repressor is still free to go and bind to the operator, switch off the operon. Sooner or later, because the cell is now lacking any glucose to make ATP, the bacterial cell is going to die. Okay. All right. Next mutation is what happens if the operator is mutated. So from here, you can already see when the operator is mutated, the binding site for the repressor molecule has changed. Okay, how can a rectangle fit into a half a circle here, right? Okay, so in this case, the repressor cannot bind to the operator, even though glucose is present. Okay, so once again, this operon is going to be synthesized constitutively or switched on constitutively. Okay, so the effect is similar to a Lac-I mutant. An operator mutant, the effect or what happens is the same as the Lac-I mutant, right? The repressor cannot bind to the operator, so RNA polymerase can bind to the promoter all the time, switch on transcription all the time. Okay. All right. Okay. So in addition to what we have seen, the Lac-I mutant or the operator mutant, okay, you also have the, remember the super repressor or IS mutant, okay, and then there's another one that we didn't look at, which is called IQ mutant. The effect is high levels of expression, okay. So maybe you can go and search yourself. How does an IQ mutant result in high levels of expression for the Lac Operon, right? Okay, so again, this is just to describe to you, all right? So in this case, I hope you know how to read genotypes, okay? So now, remember, I negative. I negative means that it's going to produce a mutant repressor, okay? Nothing wrong with the operator because it's plus, meaning that it's wild type. The Z is plus, the Y is plus, the A is plus, okay? So in this case, remember, it is constitutive expression of the Lac Operon, even if lactose is not present. Okay, all right. The next one, okay, now nothing wrong with the repressor molecule, but you have this mutant on the operator, right? So again, the result is constitutive expression of the Lac Operon, even if lactose is not present. Why? Because the repressor cannot bind to the operator. Okay? Right. What is this now? This is IS. Remember the super repressor. Okay? So what happens when you have a super repressor? Even if lactose is present, the lactose or the allolactose cannot bind to the repressor molecule because it has changed shape. Okay? So the repressor molecule can constantly bind to the operator even when lactose is present. So you have no transcription taking place. Okay? All right, so here's a summary of what we have seen. Okay, so whether it is I plus, O plus, or Z plus, of course, if lactose is present, it will be transcribed. If lactose is absent, you have no transcription. Take another example, I plus, OC, Z plus. Okay, remember OC means constitutive transcription. So whether lactose is present or lactose is absent, you will continue to get transcription of the beta-galactosidase gene. Okay. All right. So when you add glucose to the lactose, so now we're talking about both are present. Glucose is present, lactose is present. Okay. Adding glucose to lactose will reduce Lac Operon expression. All right. We saw this right at the beginning under the 4 conditions. One of the conditions was glucose present, lactose present, you get no transcription. Okay, so remember that. And if you ask a question, okay, when a bacteria cell is grown in a culture medium where glucose is present and lactose is present, is the Lac Operon switched on or switched off? Explain your answer. Okay, so you have to be able to explain that. All right, okay. So coming back to whether or not this operon is negatively or positively regulated. Okay, so for the Lac Operon, it is negatively regulated. Remember what I said? That 99%, 98, 99% of the time it is switched off because glucose is almost always available. Okay, so therefore it is negatively regulated by the repressor molecule, which is constitutively being produced. The Lac I is always there. Okay, so why is it always there? It's always there because it has to switch off the Lac Operon most of the time. Okay, almost all of the time. All right. Okay, so on the other hand, all right, remember what we also saw. We saw that when glucose is totally absent, okay, the Lac Operon must be switched on so that it produces the 3 enzymes as quickly as possible and in high quantities because it needs to keep breaking down lactose, keep bringing in new lactose, keep hydrolyzing lactose to produce glucose quickly so that the cell doesn't get sick, the cell doesn't starve because there's not enough glucose. Okay? So in this manner, it has to be positively regulated very quickly, right? So one way of positively regulating it is by doing something which is called catabolite repression. Now don't get confused just because the word repression is there, okay? Don't get confused about that, all right? So this catabolite repression is an example of positive regulation, okay? And it takes place, all right, depending on the concentration of the glucose in the cell. Okay, so catabolite repression refers to or requires both of these proteins. Okay, one is CAMP. I'm sure you know what is CAMP, cyclic AMP. Okay, the other one is CAP, which is called or short form for catabolite activator protein. Okay, and it also has another name depending on which textbook or which reference you're referring to. It can be called CAMP receptor protein. All right, okay? Okay, so how does this work, right? I'm going to very simply explain it to you now, okay? The more complicated explanation comes in a different lecture, right? But basically what you have to remember is very easy, okay? When there is a high amount of glucose in the cell, in the bacteria cell, there will be low concentrations of CAMP, okay? On the other hand, when there is low amounts of glucose, there will be high CAMP, right? Okay. Now, inside the promoter, right, the promoter for the Z, Y, and A, there is a binding site for CAMP complex 2, or this what is called the CRP molecule. Okay. So you see you have this CRP-CAMP molecule binding to the part of the promoter. The other part of the promoter is bound to the RNA polymerase. Okay, right? Okay. So when this is now binding here, in the presence of low glucose, you get high level of transcription of Lac Z, Lac Y, and Lac A. Okay? Remember or not, it's just similar to what you saw for eukaryotic cells. Remember, if you have just general transcription factors present plus RNA polymerase, okay, you will get low levels of expression, almost like housekeeping gene expression, right? But when you have specific transcription factors working with the general transcription factors, with the RNA polymerase, with the mediator molecule, then you get high levels of gene expression, okay? So this is the same thing in prokaryotic cells, right? When you have high glucose levels, your CAMP level is very low, all right? Okay, so you only get low levels. Remember, we said leaky promoter, okay? So you have very small amounts of transcription taking place that produces small amounts of the 3 enzymes, okay? But when there is not enough glucose in the bacterial cell, then this will increase the concentration of CAMP. The CRP-CAMP will come and bind to the promoter. And what it does actually is, okay, it will tell RNA polymerase, more and more molecules of RNA polymerase to come and keep binding to the promoter so that it keeps doing transcription, right? If you understand the concept of transcription, okay, you know that RNA polymerase will bind, it will move on, right? It will keep going as it reads the gene sequence to produce your mRNA sequence, isn't it? Correct? Okay, so as one RNA polymerase is moving on, now the promoter is empty, correct? So the CRP-CAMP will tell another RNA polymerase to come and bind quickly and start transcription again. Then when the second RNA polymerase starts to move along the gene sequence, the CRP-CAMP will call the third RNA polymerase, so on and so forth, so that it keeps doing high levels of mRNA synthesis for the Z gene, the Y gene, and the A gene. Okay, all right? So this is positive regulation of the Lac Operon. Negative regulation is when you have a lot of repressor molecule present and free to bind to the operator. All right, okay? So to sum up, right? So this is the structure of the Lac Operon that you have seen already, right? Your regulatory gene is your Lac I gene. You have your promoter and you have your operator, okay? The downstream of the promoter and the operator is your 3 structural genes. Number 1 is Z, number 2 is Y, number 3 is A, okay? Right. In the case where your regulatory gene or your Lac I gene is producing your repressor molecule, right? In the presence of glucose, there is no lactose. Okay, so the shape of the repressor molecule remains the same, and it will now bind to your operator. Okay, because it is bound to the operator, so it's going to block the RNA polymerase. Okay, even if the RNA polymerase can bind a little bit to the promoter, it cannot pass through this big structure here, which is the repressor molecule bind to the operator. Okay, so movement of the RNA polymerase is blocked. So you do not get transcription of Z, Y, and A, right? Okay. Now, in the second scenario, when you have low glucose levels, okay, then your permease will go and bring in the lactose, all right? The lactose becomes allolactose. The allolactose now is going to bind to the repressor molecule. When it binds to the repressor molecule, it's going to change the shape of the repressor molecule. Okay, remember, see, now it's blocked here, right? So it can no longer bind to the operator. So it is inactive. Okay, so now there's nothing here to block the movement of the RNA polymerase. So RNA polymerase will come and bind and move along the genes to produce the mRNA for Z, the mRNA for Y, and the mRNA for A. Okay, and then at the same time, you have CRP, CAMP will also bind here and keep bringing in new RNA polymerase molecules to keep moving in this direction to produce more and more mRNA, which will be translated and produced to give you high amounts of beta-galactosidase, high amounts of permease, and high amounts of transacetylase. Right? Okay. Okay. So if you've understood all that, right, you should be able, at the end of the day, you should be able to come to a table like this, all right, where we are back to our original 4 situations. Okay, remember the 4 situations we saw was what happens when glucose is present but lactose is absent, or glucose is present, lactose is present. No glucose, no lactose in the cell, or no glucose, lactose is present. Okay, so if these are the situations, you should be able to tell whether the Lac Operon is going to be transcribed or not. So we already saw from the beginning slides that transcription will only take place at high levels when glucose is definitely absent and lactose is present. Okay, all right. So we saw this part, we saw this part, right? So your job now is to make sure you can explain why. So you can explain, all right, this situation. Activator here refers to your lactose or your allolactose, okay? Repressor here refers to your Lac-I. And RNA polymerase, whether or not it's binding to the promoter, okay? So if you're given, let's say, this column and asked to fill in all this information, you should be able to do it. And you can do it not because you memorize the answer, but because you understand the mechanism of the Lac Operon. Okay? Alternatively, I could give you an additional column, right, which has Lac-I mutant, OC mutant, Lac-IS mutant, Lac-IQ mutant. Then I ask you, is there transcription or not? Right? Okay? And then I may ask you, explain why. Okay? All right? So what I'm trying to tell you is that your job is not to memorize the slides or the content of the slides. Your job is to go through them again slowly and make sure you understand the mechanism. Okay, there's a lot of information that you can find very easily on the internet. You can watch videos which will explain what I have just said again. Okay, so the take-home message is the Lac Operon is switched on only when the cell, the bacterial cell, does not have enough glucose. Okay, So most of the time, the bacterial cell has enough glucose, so the Lac Operon is switched off. How is it switched off? It is switched off by the binding of the repressor molecule to the operator. Okay, so when there is not enough glucose, how come the repressor molecule doesn't bind to the operator anymore and the Lac Operon can be switched on? So you should be able to explain that, because when there's no glucose, then the small amounts of permease go out of the cell, search for lactose, bring the lactose in, the lactose binds to the, some of the lactose binds to the repressor molecule, which prevents the repressor from binding to the operator. Then you can switch on the transcription, produce more and more of the enzymes, okay? And then keep transcribing until there is enough of glucose in the cell. Do you understand? Okay? Does anyone have any questions? One question. For leaky promoter, when repressor binds to the operator's small amount of permease still produced, then the lactose can be transported into the cell, allolactose will be produced. So how about the repressor which still bound to the operator before? That is a very good question. Okay, everything, if you see on this slide, see, all right, permanent, nothing is permanent. The binding, nothing is permanent. Okay, even when I say that the repressor is bound to the operator 98% of the time to switch off the transcription, it is not the same repressor molecule that keeps binding all the time. Otherwise, there is no need for LAC-I to keep producing LAC-I repressor constitutively. Okay, so one repressor molecule will come and bind to the operator, then maybe after a little while it will drop off, then another repressor molecule will come and bind, and so on. Okay, so in this case, in the leaky promoter case, the repressor that was originally there will drop off at some point. Okay, so then you expect another repressor molecule to come and bind to the operator and keep the operon switched off. However, that repressor molecule now has bound to the allolactose, the small amount that was brought in, so it cannot go and bind to the operator. Okay, I hope that answers the question. Does anyone have any more questions? No? All right, then please go back and read through, okay, the notes again. Make sure that you understand everything. And then this Friday, again, there's no class. So next Monday, we will look at the Arabinose Operon. Right? Okay. Thank you very much.
Thank you, Prof.
Thank you, Prof.
Okay, you're most welcome.
Thank you.